Hi, my name is James Fry. I am doing the effects of pH levels on trypsin. I am with Group C, and my lab partners are Perez, Christian, and Isis. So enzymes are catalysts made up of proteins, and they speed up reactions by breaking the bonds to lower the activation rate and therefore speeding up the reaction. Reactions would occur at slow rates and body temperatures without catalysts, but with catalysts they're sped up and able to have a higher reaction rate. In living organisms, the reaction rate would be low because the body temperature is not high enough to speed up the reactions. But with enzymes and catalysts, we're able to speed up those reactions and the reaction rate, so reactions can actually occur. In this experiment, we used trypsin as an enzyme, and trypsin speeds up the hydrolysis of peptide bonds. And specifically, trypsin digests dietary proteins in the small intestines where the trypsin is located in animals. But trypsin is first formed in the pancreas, but then is moved to the small intestines to become active trypsin, where the pH in the small intestines is approximately pH of 8. In our experiment, the question was, how will changing the pH affect the activity of trypsin on azocasein? And our hypothesis was the optimum level of pH would be around 8 or 9, and any pH higher or lower than that would decrease the activity of trypsin on the azocasein. The reason we picked this hypothesis was because the small intestine pH is around 8, so we hypothesized that the highest activity of trypsin would be around 8 or 9. So for our experiment, we tested different pHs. Specifically, we used pH 6, 8, 9, and 11. We used room temperature, which is 32 degrees Celsius, and a salt concentration of 0.15. So for our procedure, we added 450 microliters of the reaction buffer, 500 microliters of the azocasein, and then 50 microliters of the trypsin into a centrifuge tube. We then incubated them for 10 minutes, and then we added 500 microliters of the TCA into each to stop the reaction from happening. We then centrifuged the tubes for 2 minutes at max speed, and then we blanked the spectrophotometer with water, and then we tested the absorbance of each of our trials, and we read it at a 440 nanometer level. We centrifuged the reactions after adding TCA to remove any undigested substrate and make that into a pellet, and then that way we're left with the supernet, and that's what we read the absorbance of. The azocasein is a milk protein with sulfanilamide in it attached randomly, and that way we were able to read the absorbance at a 440 nanometer level. The controlled conditions used of room temperature and 0.15 molarity were chosen because we felt that was the easiest to perform our experiment. And the negative controls were prepared by not putting any trypsin into the negative controlled tube, so that way we could find out if trypsin was responsible for the activity or if it was something else. We had 3 trials for the negative control and 3 trials for each of the pHs tested, so that way we could improve variance, increase accuracy, and eliminate any error that happened during the experiment. So this is our graph. We chose a bar plot graph, and we compared the enzyme activity as absorbance measured at 440 nanometers compared to the different pHs we chose. And we chose pH 6, 8, 9, and 11. As you can see, pH 6 has the lowest absorbance rate level, and pH 9 has the greatest absorbance level. pH 6 also has the greatest standard deviation at 0.029, while pH 9 has the lowest standard deviation at 0.007. pH 9 has a normalized average of 0.14, while pH 6 has a normalized average of 0.05. This is our t-test results, and we hypothesized that pH 6, pH 8, and pH 11 would not be significantly different than the absorbance of pH 9. But as you can see, we found that the results for pH 6, 8, and 11 were significantly different than pH 9. We got a p-value of 0.005 for pH 6, a p-value of 0.024 for pH 8, and a p-value of 0.017 for pH 11. So the experimental design and execution was adequate to test the hypothesis, though I believe if we were to do the experiment a third time, we would get even better results than the results we got with the second time we did the experiment. And with our experiment, we found that our hypothesis was partially supported. We hypothesized that the optimum pH level would be between 8 and 9, and we found out that the optimum pH level would be pH 9. pH 9 had a normalized average of 0.14, which was the highest normalized average out of all the pHs we tested. This means that the pH 9 had the highest activity rate for trypsin on azocasein. As stated before, for the t-test, all of the different pHs compared to pH 9 were significantly different. The lowest being pH 6, which had a p-value of 0.005. So other groups also found that the optimum pH would be pH 9, and similar to our experiment, they had control conditions of room temperature and a salt concentration of 0.15 molarity. We had a room temperature of 23 degrees Celsius, but the National Institution of Health suggested that a optimum room temperature would be at 37 degrees Celsius, which may have increased the activity of trypsin on the azocasein. Two sources of variability could have been that once the incubation period was over, we did not put in the TCA fast enough, and that could have increased the activity since the reaction didn't stop in time. Another source of variability could have been that not all of the undigested substrate was put into the pellet when we centrifuged, and that means our absorbance rate would have been off when we measured the absorbance. Improvements to this experiment could have been a warmer room, so maybe the activity could have increased, and maybe more practice with pipetting since it was our first time using the pipettes. Our conclusion that the optimum pH would be pH 9 suggests that the optimum ideal conditions to grow bacteria would also be in the pH 9 range. So that way the highest activity of trypsin on azocasein would be at its highest point. Thank you for watching.